Welcome everybody to our external webinar. At today's presentation, we are going to have Dr. Marianne Martoni. Dr. Martoni is the principal investigator of the Neuroinformatics Framework project. She just completed her tenure as a U.S. scientific representative to the International Neuroinformatics Coordinating Facility, where she still heads the program on ontologies. She is finishing up her tenure as president of Force 11, an organization dedicated to advancing scholarly communication and e-scholarship. In today's presentation, she will be talking about hypothesis, creating a lightweight, open, portable knowledge layer over biomedicine. Please welcome Dr. Martoni.
Thank you, Rosemarie. Can everybody hear me and see my slides?
Yes.
Okay, great. So I was just on a webinar, I think it was 2 weeks ago, to talk about the Resource Identification Initiative with my colleague, Dr. Anita Brandowski. But today I am speaking in my capacity as the Director of Biosciences for Hypothesis. Hypothesis is a nonprofit company that is developing a technology to do exactly what the title says, create a lightweight portable knowledge layer, essentially over the web, but over biomedicine in particular. So the mission of Hypothesis is really, again, to make sure that the web has a layer that originally was envisioned for it when people conceived of the web. That is, that it was an interactive thing and that you should be able to take a document and highlight things and add an annotation to it. For various reasons, this never was realized at the time, technology and other things being what it was, but it is now possible to do it. And for those who are interested more in the history of Hypothesis, there was an article that just came out in Nature on annotating the scholarly web that can give you this information. So annotation is distinct from commenting, and that annotation, as you see here in this book, is very much tied to individual phrases, individual pieces of knowledge. So while the web has commenting systems all over the place, first of all, they are a very small part of the web. They are controlled by the people who create the content. They have to deliberately embed the comments into it. They are sort of at a scroll down at the bottom. And in many cases, they may be in a proprietary enclosed format. That is, if somebody goes to one site and comments on an article, and that same article is in another site and comments it, the comments don't transfer. You also basically need a different account for every site if you want to go around the web and leave some comments. Of course, we don't have to do that. We can do what we do now, which is we go to different places and we add content, or we comment on an article through a blog or some other reference form. Even in a scholarly article, we refer to something that is at a distance. So essentially, it doesn't really have to be like this now. With a web annotation, one can actually create an interactive layer over the content as it appears. So it works as a layer over a web page and a PDF. Right now, it primarily works with text, but as I'll show you, images, video, and other types of research objects are coming. Hypothesis was founded as a nonprofit because they believe that this technology should be ubiquitous, open sourced. It should be standards-based, that is, interoperable. There is a mission here to enable this conversation, and that it should be easy to use. So essentially, we are at the cusp of developing a capability for the web that should be just like the web. There is a working group in the W3C, the authority that defines standards for the web, that is looking at web and creating a standard for web annotation. But more importantly, any compatible client should be able to create them. Any server should be able to deliver to them. A client should be able to listen to whatever sources of annotations they want to. So it's not that if I create my annotations using Adobe Reader and I need to use Adobe Reader to read them, it should be portable and interoperable. Nobody has a control over them. And I think that this brings a very sort of profound set of questions, particularly for science. But here it is more importantly, no one has a monopoly over the format. It belongs to everyone. It's an open format. And the software and the service providers are not locked together. So essentially what this allows us to do is to have an independent layer over the existing web that is not controlled by any one vendor, any one publisher, any one information source. So before I go into specifics for biomedicine, I just wanted to show a little bit about how Hypothesis works. So I have a little video here, and if there are questions, I can always switch to a live demo. But essentially, I'll show you how to install Hypothesis, how one annotates it, some of the functions that are currently available. So this is our home page, and let me click it, Hypothesis, in order to get there. And in order to use Hypothesis, it's free and open source. You can install a plugin, or you can, let me just pause this for a while, you can add it to your own website. So you can embed it into any browser. There's a plugin for Chrome, an extension for Chrome. There are also ways to use bookmarklets for Safari and Firefox. But also using just a little coding, you can make it natively available in your website. So you're able to embed it into your website. It does require an account, a free account, because these annotations are tagged to you, so we know, who is making the annotations. And you can sign up on the website. Unlike that book that I showed you that had all the underlines and the post-its and the fact that we were all taught when we were kids, at least at people my age, not to write in our books, these are layers, so you can turn them on and off. So if you see the sidebar here and you activate them, you can see that there's highlights. If you pull out the sidebar, you can see the comments that people have left on that page. You can also collapse it and turn it off, so it's non-destructive. It doesn't have something that you need to actually do. So let's watch how this actually works. So here I'm just going to our blog, highlighting a piece of text, and a little box comes up, and it lets me essentially type in an annotation. Again, this is not a comment. You notice that it is tagged to the specific piece of text right here, and that the sidebar, this number, actually corresponds to where that annotation is. So it appears in line or close to in line with the thing that you're referencing. Because Hypothesis is geared towards the web, you can do things like insert links. So here you can see we can create a link back to Hypothesis using just a little markup. And you can also add your tags, so you can tag any one of them, which makes them searchable later on. Let me just go back a little bit. The other thing is that annotations right now can be both public and private, so you can annotate for your own purpose and make these annotations public or private. And we've also added the capacity now to add in private groups. So if you and your colleagues want to annotate together, you can create a group, share the link with them, and only that group can see them. So it's very flexible in that way, in that not every annotation is meant for the public. In this case, I'm posting it to the public. Again, you can turn them off and on and see the highlights. You can share annotated documents even with people who do not have a Hypothesis account. There is a proxy service called VIA, and it is activated by this link up here. Essentially, if you send that link by email to somebody, they will be able to see the annotations on top of a document, although they won't be able to reply to them or add their own annotations unless they have an account. Get going again. So again, these are sortable. You can actually search them. We have a stream. Everybody gets their own annotation stream that's tagged under their accounts, and you can use tags and other things to be able to search across your annotations, across all documents, not just the one you're in. There's even a public annotation stream, so at any given moment you can see conversations that are going on in the web around the world in different contexts. Of course, I know most people are here today because of the possibility of using Hypothesis in the sciences and using annotation in the sciences. Here you see an article in PLOS that has been annotated. Whoops, let me go back. Let me get back here. Sorry about that. Here's an article in PLOS that has been annotated, in this case by me. And as I said, these are geared towards the web, so you can reply to an annotation. If you do that, the person who made the annotation gets a notification, just like if you're used to working in Google Docs. In this way, we're able to create these interactive forums around articles that were heretofore static. But again, because this is a layer, you can turn this off or on those who created this document do not need to be disturbed by the things that people are saying about them, but those who want to have conversations around them are allowed to have them. In this way, we can take static scientific artifacts and we can turn them into interactive forums. So how is this being used in the sciences in general and biomedicine in particular? Many people will say to me, well, you know what, that we have commenting systems out there, people don't use them, why should we, think that people are going to use Hypothesis? And I point out that actually, again, annotation is different than commenting. But more importantly, if you think about it, biomedical scientists annotate every day, all over the place, all the time. And not all of those annotations are the type of conversational layer that many people in public engage with in Hypothesis. Some of them are just personal notes or thoughts. You read articles, you highlight things, you organize your notes. We do, however, a lot of knowledge extraction, right? So we have a lot of people who are reading the literature, they're doing entity tagging, they're taking information out of the literature. It would be great to have, again, an open source set of tools that provide this capacity in this very web-enabled interactive way so that everybody doesn't have to rebuild all of these tools, and I'll show you some examples of that. We also do a lot of knowledge addition. So annotating is, of course, standard practice if you're doing gene sequences, imaging, linking information, all of that stuff. We add human knowledge to parts of data all the time. And I created this little cartoon to just show the number of conversations or knowledge additions that happen in a typical biomedical workflow. I used to be an electron microscopist, and right from the very beginning of conceiving the experiment, there are notes that are attached to your lab record about how well something went or didn't go. We create knowledge. This is a neuron. We add information when we segment it, we analyze it. We make more statements about the fact that the things that I see are normal or abnormal. We go through a publication process where there's a back and forth between the authors and the reviewers and the editors about information in the paper. Why didn't you do this? Oh, why am I accepting this? After it gets out into the post-publication world, again, a lot of conversations, knowledge extractions, things are going on, and more importantly, in that sort of post-publication world, we have some of the content that's walled off over in Nature, we have some of it over in Wiley, we have some of it in Elsevier. Even within a specific article, it is read in different places. So knowledge is in fact added at all steps. But right now, it's non-transferable. Everything is done in a proprietary system. There's no way for the knowledge to sort of port over. There's no way for these conversations to occur. That's really what open annotation on the web starts to allow us to do. So how is it that Hypothesis is currently being used in biomedical science? I'm going to go through some examples, but I'm hoping that you can see that based on that previous cartoon, that across the entire scientific workflow, there is a need for this open knowledge layer that is able to connect together all the different tools, places, and people that will touch a piece of data or an article over the course of its lifetime. But obviously, one of the biggest focuses of Hypothesis is what you can do with something after it's published, so post-publication review. We are having it being used in journal clubs so that people can interact over a document. Large educational program. I think you can see that there's a lot of educational opportunities for being able to use open annotation. I'll show you some examples of scientists who are annotating their own articles just to update information on them, because as soon as we publish articles, they tend to be obsolete. We have some examples of people trying to do that very structured type annotation, which is very familiar to people who are curating the literature. We have a mandate now from NIH and others to make our content more accessible, so we'll show you how people are annotating articles to make the content more accessible. And more importantly, we're now starting to work with the Neuroscience Information Framework, my colleagues at UCSD, to allow Hypothesis to be able to take automated annotations that have already been generated and are kept in a database, and be able to project those into the article so that people reading an article can take advantage of this information. So here's just a few examples of the sorts of things that are going on. Hypothesis is a very quick and easy way to not only take notes on published materials, but also to provide feedback on published articles. So we had a little project with Wikipedia where we had some scientists read Wikipedia articles, and rather than having to learn how to natively edit the wiki, they could just very quickly provide feedback on information that's inside of those articles. This is Wikipedia. One can imagine the same things happens with the scientific literature. One of the most spectacular use cases of this is actually a group of scientists, an organization called Climate Feedback. These are climate researchers who read news stories on the web. As you know, climate change is a very controversial topic. And they will read articles that appear in The Guardian, The Times, The Wall Street Journal, and add annotations to them. They make these annotations publicly available, and they rate individual statements. They'll link to additional evidence. They'll say if there's counter evidence. And what's interesting is that based on this annotation, there's actually been some corrections that have been issued in a couple of articles about climate change. So it gives scientists a voice on what's going on in the popular media. One of the most exciting things, again, I think, is that in a very non-destructive and interactive way, as I mentioned, it turns research articles or other research objects into living documents. So this is Alyssa Chesler, who's a scientist at Jackson Labs, who actually annotated one of her papers to provide updates. So, for example, she points out that the GitHub Community Code repository that was mentioned in the paper, which was published back in 2012, is now in Bitbucket. They've moved it.
Excuse me, can I interrupt for a second? I'm sorry.
Yeah.
Can you please mute your phone? There's very noisy background.
Someone is making a lot of noise, so please mute your phone.
So I invite people, in fact, to go and read some of their old articles, as I have done, and I was surprised at the number of things in there which have been updated, abandoned, migrated, or moved. And if you would like people to take full advantage of your papers, it's a really easy way for you to put that additional information in, because the publishers aren't going to be able to do that or are not going to want to do it, and it's going to constantly change. So it's, again, very lightweight and very easy. It takes 10 or 15 minutes for one to go through one's article. As I mentioned, it's a really good way for making your science accessible. And in fact, we have a group of graduate students with the American Association for the Advancement of Science who are going through science articles and using Hypothesis with some customized interface components that they've made to make these articles more accessible for use in the classroom. It's called Science in the Classroom. So they provide glossaries and links to previous works, authors, experiments. So it's a very powerful way to make one's science more accessible. Again, it opens up communication channels around scientific articles where one can ask questions. So as my capacity working for Hypothesis, I was looking at various scientific articles that had been annotated, and I saw one where somebody asked a question about gene networks versus brain networks, and that was 6 months ago. I answered it, and he replied, "Thank you very much for giving me this answer." So one can see that you can set up communication channels with authors and other people around one's paper so that we can provide additional information, additional clarifications that were not possible before in a very kind of asynchronous, atemporal way. So people can ask a question, people can answer it when they have time. Here's that little exchange. Hypothesis in the Classroom. Dr. Jeremy Dean at Hypothesis has a very active program in education. This fall, there were over 50 classrooms that were using Hypothesis, including Dr. Elba Serrano at New Mexico State University, who had her graduate students reading and annotating on neuroscience articles, as you can see right here. So many educators are finding, again, that this is giving a new capability, a new layer to students as they are able to collaboratively read, collaboratively feedback, work together in teams a lot more effectively. And as we know in science, science is moving towards teams. This provides a very easy way for people to get together and share the reading experience, something that I think is very, very important in STEM education. So Hypothesis is becoming part of a flipped or blended classroom experience. As I mentioned, a lot of some of the use cases we're seeing in biomedicine are less about this sort of conversational layer, because many people are like, well, I don't want to do this in public, I don't want to do this, and I don't want people to see. That's fine. Hypothesis is just a very useful general purpose code base and toolkit for doing the types of things that we typically associate with annotation in biomedicine. So this is a use case by my colleague David Kennedy, who I think is on this webinar, from NITRC, the Neuroimaging Tools and Resource Clearinghouse. And he has been using Hypothesis as a quick way to produce reports by tagging the literature, essentially inserting a little bit of markup into the annotation. Because Hypothesis is free and open source, you own your annotations, you can get your annotations back, and he was able to export his annotations into a tabular representation, essentially to do that type of entity tagging, that structured annotation that biomedicine does all the time. But again, there was no coding that was necessary. There was no new code that needed to be developed beyond very simple import and export routines, or simple markup and export routines, saving everybody a lot of time and energy. We have been hearing from people who do biocuration that this would also allow curators around the world to see what information has already been extracted from an article, so that it doesn't need to be done twice, and other people would be able to take advantage of the work that's going on. For those of you who were on the webinar, I think it was last week, on the Resource Identification Initiative, you know that through Force 11 and NIF and some of the other projects I'm involved in, that we have been working to get a new type of annotation, essentially a citation into the literature called a resource identifier. Essentially, authors are supplying accession numbers from authoritative databases that serve to identify key resources that they used. Those are right now antibodies, genetically modified animals, and software and databases. And there again, NIH has established registries for these entities around the world. If you know about these registries, you can go to them. But we were finding that in the literature, most researchers were not providing enough metadata for us to be able to identify what they used. And so the solution was, give us an RRID, this accession number that comes out of this database, make sure that it's outside the paywall, and make sure that the syntax is uniform across journals and publishers. And if you heard the webinar last week, you know that this has been a very highly successful pilot project. We've got over 70 journals participating, and these RRIDs are appearing inside of the literature. Right now, these RRIDs don't do anything. There is a group at Force 11 that is working on making them machine resolvable, but essentially, because an RRID is a regular expression that doesn't take very much for a text miner to be able to identify, we are looking at working with NIF to use Hypothesis to be able to easily resolve these RRIDs and attach different information to them in the context of the article without having to send people to other sites, without the publishers or anyone else having to develop any special machinery. So this is a pilot project right now. It's in its first stages, but essentially we have a little piece of JavaScript that on any article will go and try to recognize RRIDs. It will highlight them. You can see that in this paper it found 5 antibodies and 2 tools. It then imports information automatically from the NIF SciCrunch database about these RRIDs, and they're piped automatically into Hypothesis. So this next movie actually shows that. This is a Journal of Comparative Neurology article. We've run the script already because it's a little slow since it's just beta code, that found animals, antibodies, and tools. And if we now make the movie go, you see that right now there's no annotations there. But if I click on the little RRID resolver and then I scroll down, now you see that an annotation has appeared. And it is highlighted, and if I open that up, it has pulled information from the RRID database and provided additional information about that zebrafish in this case. And this was all done automatically. So this is essentially a machine-generated annotation, machine-generated entity recognition put together so that we can now project this information into papers in a very flexible and dynamic way. And I have to say that this was one of the things that attracted me to Hypothesis when I first learned about it, was that those of us who are running data repositories, resources like NIF, we know that many people don't even know that they exist. And essentially, if you talk to informaticians, they'll tell you that researchers spend their time in two places, Google and PubMed. So what I liked was that Hypothesis actually was a way for us to project NIF's information that we have into the place where people are reading it, rather than having them required to know that we exist and come over there and look at it. So again, it's a lightweight, portable knowledge layer that can connect the different pieces of the ecosystem without requiring a whole lot of new development on the part of either the developer, the people who are providing the content, or those who are creating these resources. So essentially, I think also that beyond being simply useful, this really gives us the capacity to do something very new and very significant. We all have been hearing of the reproducibility crisis in science, in biomedicine in particular. We know that part of that is that information transfer is very, very slow and ad hoc. So people can be reading about a paper that references a cell line or an antibody. Somewhere in those 28 million articles, there may be a statement or a paper that says, oh, hey, you know what, this doesn't seem to work very well under certain circumstances. We know we're encouraging people to put methods into their papers that are very, very detailed, but we also know that those are not perfect, and that allowing people to put updates and start conversations about these methods would also go a long way towards making these things more reproducible. We also know that the authors themselves, who care very much about their work, would like to be able to provide updates and what have you, but trying to do that again more formally through these commenting systems, it's just not, it's difficult to make comments, and they're so divorced from the material itself, locked into that one system, that they don't transfer. So again, if we sort of think about this, what if over any paper we could know if the paper's been retracted? Oftentimes that information's not available right there when you're reading it. What antibodies are being used? Have issues been reported with them? Who else has tried to reproduce this? Are there additional notes by authors since the original publication? What do my peers think of this? What Hypothesis gives us, open annotation in general, Hypothesis in particular, is that this actually makes this possible now. And we are actively working on putting the partnerships together to be able to do things like you see right here. I recognize that an RRID is there. I know that a validation report has been produced over in another place, and so when one is reading about that antibody, you can find those validation reports. You can find links, additional information that is available for any given research resource. So even though Hypothesis is sort of simple in the sense that it says, well, we annotate, we take notes, I think the implications for biomedicine are actually quite profound. So what are the goals of Hypothesis? We've talked mostly about text, but this is BioCaddy, which is the Data Discovery Index. And I actually think, and we've been actively working with my colleagues who have data repositories and other types of things, computational platforms, software platforms, to recognize that actually open annotation, as I showed you in that cartoon, should be a part of anything that we do. It should be a part not just of articles, but it should be parts of databases, those that provide data sets. Any place where people are looking and using information, there ought to be this feedback layer that's possible. So we're starting, as I'll show you at the very end, a very ambitious project called Bringing Open Annotation to All Research Objects. Data annotation is a major use case in biomedicine. Everybody's got to build their tools from scratch. Everybody builds a proprietary format for that particular tool or context. We do not believe that the algorithms that would recognize a gene sequence or a particular entity inside of a paper or a part of an image are a part of Hypothesis. Those are the science that everyone needs to develop on their own. But as you saw in the RRID case, there's no reason why one cannot link that type of algorithm to an open annotation framework so that annotations can sort of pass through, they're web-enabled, they're searchable across contexts, all the things that web annotation allow us to do. I also think, again, in very practical terms right now, having run a data repository, there's a whole lot of things that go on, just like with journals. There's a back and forth between the people who submit the data and those who might curate the data. They ask questions. How nice is it, instead of having to do all that through email, to just be able to open up a private conversation with the two parties, highlight specific phrases, ask questions about those particular things, and get feedback from the author. So we are, in fact, launching a project, integration of Hypothesis into all data repositories, to let people know that these tools exist and to see how they might be able to use them. So I think my colleague Russ Poldrack up in Stanford, he is with a bunch of other neuroimaging colleagues, have a data sharing site called NeuroVault. Hypothesis is very easy to install. Essentially, a graduate student was able to install Hypothesis in less than a day and use it to provide a metadata curation tool. They essentially need to read the papers that come along with these data sets, and they extract various quantities from the literature. And she was able to sort of set that up right here in part of a workflow so that people could use Hypothesis to provide that information. They could extract it and insert it into their database. Again, Hypothesis works with any web-enabled object. So you can right now, if you have a CSV file on the web, you can annotate it and ask questions, highlight specific rows and entities. This is a very nascent capability. We're seeking partners to help build this out. But again, we can broaden out the things that we annotate, not just to narrative and text, but also to data files and then data objects themselves, like sequences and imaging, and think again how the Hypothesis interface and services can be used on top of those. We are very pleased that GigaScience, which many of you may know, that is both a journal, but they also have a database for putting the data associated with papers into making them openly available. And GigaScience has just integrated Hypothesis into their database. Right now, it's mostly working with the abstract pages, but the actual data sets are soon to follow. Again, it is very easy to integrate this into any given database, and therefore it provides, again, this very sort of lightweight, cost-effective means to adding this capacity to any scholarly object. So what's on the horizon and what's next? I've shown you some of the use cases for Hypothesis, the way that people are using it now. I've shown you some of the pilot projects that we're working with on various partners to make Hypothesis more useful and integrated into the biomedical workflow. I'm sure many of you have questions, and I'm going to leave plenty of time for questions because this is a new capability. People ask about moderation, and they ask all kinds of things. So there's several things on the roadmap that we think are going to be important to the scholarly use cases that biomedicine may want to address. One of the very first ones is that we are going to be integrating the ORCID ID, the researcher ID, into annotations. So authors will be allowed to tie annotations to their unique author identifiers. Why do we want people to be able to do this? There's really two reasons. One, we want to give authors credit for annotations. So in domains such as the humanities and in other areas, an annotated document is considered a scholarly work. The annotated works of Shakespeare, the annotated works of Byron is a work of scholarship. And you can see that the science in the classroom, you can see that some of the deeper commentary that people are providing over articles are works of scholarship, and they should be credited. So we want to make sure that authors are getting scholarly credit for annotations. But more importantly, if people are allowed to say things in a public place about something that has been created, we'd like to be able to evaluate the expertise of the annotator. So if by linking to their ORCID profile, you would get some sense of who it was who was saying this. It would also allow us even automatically to perhaps be able to say this might be a competitor because they publish in this domain. I think there's all kinds of things that can be done once we know who it is that's annotating. We also want to be able to surface role-based annotations, and so Alyssa Chesler annotating her own paper as an author of that paper may be evaluated differently than somebody just coming along and asking a question. An editor may choose to add some editorial comments to a published article because they knew something about the peer review and the compromises that were made to get the paper into the scholarly body that they may want to be able to put in.
Is everybody there?
Editors of special issues, again, may want to read and put things across the different papers in there that let them be tied together. And you would like to know that the editor and the author are the ones, in fact, who are doing these annotations. So tying to the ORCID profiles allows us to do some of these things, and this basic capability is planned for the first quarter of 2016. We're also working with altmetrics. For those of you who know altmetrics, that there are other things that guide how much of an impact a scholarly object has. Are people reading about it, or blogging about it? Are they downloading it? Are they tweeting about it? We also think if people are annotating it, whether they're annotating it because they like it or don't like it, that's part of their impact, and that should be part of the altmetric score, and we are actively working with altmetrics on that. The next thing is that there's many formats. So right now, one of the capabilities is, in many cases, if you annotate the HTML version or the PDF version of the document, provided that the PDF has been produced in a way that allows us to create document equivalents, those annotations will transfer. So whether you've annotated the PDF or the HTML version, they will transfer. But there's also e-publication formats. There's other sorts of formats that need to be addressed because nobody is going to be reading the same document in the same format in the same platform. And the whole purpose of Hypothesis is that we can help transcend this environment. We are also working to integrate into popular apps such as PDF readers and book readers. There's some functionality on tablets, but it doesn't work particularly well. I showed you some examples of the structured annotation use case, which is very important to biomedicine, and David Kennedy has been a great sport in testing this out with us. But these are prototypes, and we know that this needs to be built out a little bit better. One of the very first functions that will be coming within the next few months is the ability to tag with controlled vocabularies. So right now you can tag, but those are user-supplied tags. We would like to be able to tag with ontology terms or whatever vocabulary somebody chooses to sort of enforce structured tagging. And we also are planning on having an annotation dashboard to make it much easier for users to be able to manage their own annotations. I myself now never read the web without my Hypothesis plugin activated because I just find it a very useful way to organize things that I have been reading. But our biggest initiative is this annotating all knowledge. So this was the initiative that was actually featured in that Nature article. And Hypothesis, again, was founded, as I mentioned, as a nonprofit because, of course, we want everyone to use Hypothesis. But it's more important that in the dawn of this new capability, this interactive overlay on top of web-based objects, that we truly engineer that for the 21st century and avoid some of the silos and things that have come up in the more traditional forms of transferring scholarly content to the web, where everything is locked across these different platforms. We really want annotation to be developed as an open capability. And therefore, we've been contacting and working with major partners. So you see Wiley and ORCID Ubiquity Press, HathiTrust and JSTOR, eLife individual journals, to essentially create a coalition that can help define both what this knowledge layer is going to look like and how it is going to interact and operate, that is, the norms in which that we develop to actually use it, but also that everybody commits that this is not something that's going to be locked down, that it is something that's going to be able to be transferable and non-proprietary. And we've been very surprised, or I have been surprised, maybe my Hypothesis colleagues haven't been, at how much people inside the publishing houses understand that this is in fact the way that it should be done. That this is, I think, the first time where everybody has been willing and able to kind of come together, and we hope NIH and all of the funding agencies do as well, that say this is a capability that should be truly engineered for the web, and that it is not owned by any individual. They may create good tools on top of it. They may create custom instantiations, but that they are declaring that we are not going to own those annotations. Those annotations belong to the people who created them and essentially the people who created the works on which they reference. So this is actively building up. We're going to have our first meeting at the FORCE 2016 meeting in Portland, but I think we really want to be able to make sure that that we get through to the major publishers, major producers of scholarly content, places like ResearchGate, anywhere that scholarship is created, to say that we have an opportunity to create something that connects across all of scholarship, and we want everybody to join. And we want BioCaddy, and we want NIH, and we want everyone else also to sort of join this effort. This is not a declaration that you will use hypothesis. It is rather that you are committed to creating this non-proprietary portable knowledge layer and incorporating it and making it ubiquitously available across all the different objects that we create. So I would like to thank the Hypothesis team. They are actively growing, so this is already obsolete, but particularly Dan Whaley, who was the founder of this, and John Udell, who's been working very closely, our product manager, with my biomedical colleagues to create these demonstrator projects. But it is an active and growing nonprofit. We're also grateful for funding coming from the Helmsley Foundation, which is specifically supporting the biomedical program, but also the Mellon Foundation, Knight, Shuttleworth, and Sloan Foundations, who have been instrumental in getting it up and running. And these are some links to recent press articles if anybody is interested in them. So I wanted to make sure that I left plenty of time for comments and discussions or live demos, whatever it is that people might like to see. Thank you.
Has anyone a question?
Marian, this is Hua from UT Health.
Hi, Hua.
Great talk, and
Thanks.
It's very interesting tool. I'm just wondering, what are the license for Hypothesis? You said it's open source, right?
Yes.
Any particular open source license?
There is an open source license. It is, I think, the most permissive one that is there.
Oh, okay.
Yeah.
Like a Apache 2 or something like
Right.
that.
And the annotations themselves, the public annotations are CC0. So private annotations are private, but any public annotation is a CC0. That is, they are meant to be reused. Mm-hmm.
So right now it's only working for the browser type. Are you guys also planning on like a mobile device version?
Yes. We get that requested a lot, and that is on the roadmap.
Okay. Thanks.
Uh-huh.
Marianne.
Hey, Marianne, this is Julie here. I just wanted to say I texted you some questions. I apologize. I'm in a very noisy environment, so I'm going to turn my mute on. I'll let you read the questions.
Oh, okay.
You don't have to answer all of them right now.
Let me look at the text. Ron, while I'm reading those, did you have a question?
Yes. Let's talk about, as I understand this, the annotation there goes into publications. And what about...
It goes on top of publications, yeah.
On top of publications. Okay. What about data sets?
Well, that's what I said. Right. So right now, if you have... So the Hypothesis and the Open Annotation Standard, there's been some work on an Open Annotation Standard for images to attach annotation to parts of images, but that is not fully enabled yet. But essentially, I showed you the slide with the CSV file. Any web format or things that can be converted into a web format, you can annotate. What you can't do with images right now is attach an annotation to a specific part of an image, but you could highlight the figure legend, for example, and annotate that. The data uses are the ones that I'm most interested in exploring because I think, again, an open annotation toolkit that can be delivered to biomedicine to handle the most common types of data objects is a very important thing to have. Images, I think, will be one of the first ones, but also tabular data. Now, there are some questions. People often ask me about dynamic data. What happens if the data goes away? What happens if the Wikipedia article changes, right? The web is a mercurial place. And so there is some fuzzy anchoring, so that if the thing that you see is still around, we tend to be able to find it. But if it goes away, there's now a type of annotation that's called an orphan annotation that says, well, this was there. The snippet is captured, so you're able to see what it was that was annotated, but it's not there anymore. And I think, again, because this is a new capability, the conventions around how these things should be that are going to be handled will start to emerge, because obviously those orphaned annotations belong to an earlier version of something, and so it's part of the historical change document. We are working with the Web Archive so that at some point when you do annotate a document and hypothesis, you'll be able to save a copy of it and its annotations to the Web Archive. But one of the use cases that we're starting to explore with partners is dynamic data, right? So the data changes. I can go into a NIF and issue a query, and I can annotate the result set that comes from that. I actually do that all the time because I find it a useful tool for organizing things as I explore data set to data set. But if a new dump comes in and that data element is now ranked 50th instead of second, will it find it? Those are the sorts of things that we need to explore. But I think, you know, the goal of hypothesis is to make sure that there's an open annotation layer for all things that we want to annotate, and in biomedicine, data is one of the big ones. Okay, let me read Julie's thing. So the W3C, are we using their open annotation model? We are on the working group to define the web standard for the W3C. It is on the roadmap to actually port the annotations to the annotation model, but we are not using it yet. That's what often happens with concurrent development. For this to work reliably, the identifier's landscape has to be a whole lot better. What is your plan for volatile content, content that doesn't persist? So I talked a little bit about that, but I think it also starts to point to the big document equivalence problem that we have. And again, when the web was first, when the publishers and others came to the web, we started to say, oh, I can sort of splash this thing up. But if I want to create an ecosystem, right, the fact that the HTML version, the XML version, the PDF version all have different identifiers, they don't map to each other, and that a publisher may not even put in the appropriate header that says, oh, this actually maps to this HTML version, you actually made the connection. So what we start to uncover when we do things like this are some structural problems with the way that we do things now. The good thing is that they can be solved when people start to think about being part of an ecosystem and a network as opposed to what I'm hearing now, the term vertical, right? So I'm the vertical, you come in and you're going to stay with me and you're not going to go anywhere else. We're starting to see where just simple changes in practices to make sure that this document equivalence occurs. Once that happens, it's very easy for us to sync across the two modes. Dynamic data and that, again, when the annotation, the thing that it's anchored to anymore, is not there anymore, it's considered an orphan annotation. What we're starting to do, we have a designer, Connor, who is saying, well, there are different types of annotations that are attached to a page. We should at least let you know that these things were there, but they're not anymore, and through the Web Archive, understand what was there at the time. Is the annotated text available publicly or just the annotation? The snippet that is annotated is available publicly. And this question about maximum word size, so she's asking if there are maximum word size that the paywall publishers are authorizing, and yes, there are conventions around that. I don't know if it's a formal legal binding thing, but right now you are able to display these snippets, but good annotation behavior suggests that you should not be able to use annotation to break paywall. In fact, right now, because of the document equivalence problem, that the abstract and then the paywalled article have a different URL. If you've annotated the paywalled article, unless you have a subscription, you won't even be able to see the annotations. We don't think that, unless you look in the stream, but we think, again, that you should be able to see the annotations independent of the paywall. So let me skip to somebody else because you had a lot of questions. And Mary Mangan said, is there a way to delete offensive or spam comments? Right. So moderation is one of the big questions we get all the time. And there is a nascent capacity to be able to delete. There is a capacity for hypothesis right now to delete offensive comments, and there's a code of conduct. Fortunately, we haven't had to do that. But the question becomes is who is going to moderate this? And that's one of the reasons the group function was put in, because one could conceive that individual sites would have moderators that would look at this and automatic spam detectors. But as I said at the Society for Neuroscience, where I sort of debuted Hypothesis to Biomedicine, all of these things are going to have to be thought through. That's why we formed this coalition, and we really, again, are at the beginning of a very new capability. Fortunately, there are some things we can learn from other communities, so the capacity to say if you're going to annotate in public, there are rules of conduct, we can flag offensive language, but we're never going to be able to stop everything. That's why moderation is so important. Hypothesis, however, really has marketed itself, if you will, to the scholarly audience. So there's other companies that are dealing with more popular content. Hypothesis has really geared itself towards scholarship, and thus far everybody's been behaving very nicely. But moderation is one of the key outstanding questions, and this is why we want to get this into people's hands to find out what it is they'd like to do. We do feel that a journal, for example, that embeds Hypothesis ought to be able to say, this is our authorized channel. Right? It's not going to stop anybody else from doing what they want to do, because right now you can say nasty things about any journal article on the web someplace else, but that you would be able to say this is the official journal channel and turn off all the other channels is something that I think is in our planned roadmap. If someone publishes their annotations to the world, do they relinquish their ownership? So I think I said yes, they're CC0. So that's another question that people need to discuss. But right now, everybody is allowed to use them. I think that they should be searchable. I think that they should be, whether it should be CC0 or not, or CC BY, I think these are questions, but they are CC0. And I think that that was deliberate. I don't want to speak for the rest of Hypothesis, because so many sites that take comments and things own your intellectual input, and Hypothesis did not want to exert any control over what people annotate. It's theirs. Okay, I think I got most of them. Where are the public annotations stored? So that's a good question, Peter. Hypothesis, you can establish your own Hypothesis server. That is the way that the architecture was originally designed. However, just like you can have your own web server, all you can use our Hypothesis cloud service. So most people now are using the cloud service. Hypothesis, even though we've expanded lately, like most nonprofits, is small. So despite the fact that there are, that it is always intended that people be able to do this on their own, sometimes you just have to be practical. And for this phase here, where we're trying to let people know what a web annotation is and how it works, we've really put a lot of effort into our own service. So that is the one that most people are using. But we do have people who set up their own servers and control the content. As I put in that slide in the beginning, it really is meant to be like the web itself. You can run your own web server, you can use a cloud server, it's really up to you. And there are some people, again, so in the peer review pipeline, they're not using the Hypothesis interface, but they're using some of the code in the back end to create that service, but they're creating their own interfaces around it because it's a much more specific and detailed workflow than what the simple Hypothesis interface can implement right now. So we really are trying to be as flexible as possible, but also, again, encouraging people to sort of join now in this effort so that these things can be engineered and used in the best way possible. Okay, other questions?
Mm-hmm.
I think I got most of your, oh, there's another one. What kind of setup is required for a publisher or data to enable Hypothesis? So embedding it into your site is just a little bit of JavaScript that you put in, and then Hypothesis just appears in that sidebar. People don't have to install the plugin. And for most sites, that's very easy. There is a WordPress plugin and also a Drupal plugin that people can use to make it available through those sites. We do have a product manager and others who can work with you if there's any troubles, but generally I've been hearing that it is relatively easy to install. We don't yet sync with local user accounts. That is, again, on the roadmap for the coming year, so that if you log in, you'll be able to log into Hypothesis at the same time. Right now, you need your Hypothesis account and your local account if it's a password-protected site. But installing Hypothesis itself, there's that place on the website that says add it to your website, and it will take you to the code base. There's an open GitHub repository, whatever you need. And the Hypothesis team is really great about providing user support.
So, all right. So I'd like to thank Dr. Mattoni for this great presentation today. The slides and the recording for this webinar will be uploaded on the BioCaddy website at biocaddy.org later on. Our next meeting takes place on January 14th, 2016 at 10 a.m. Pacific Standard Time, and the speaker will be announced later on. Thank you, everybody, for attending this meeting.
Yes, thank you very much, everybody, and please feel free to contact me if you have any questions.